right, hello. Next part, let's talk about muscles and shells. So hard parts and the things that move them around. All right. So this is like a long ways slice down the middle of the brachiopod. You can see our pedicle coming out here. And so this valve that's on the bottom that has the pedicle coming out of it is, you guessed it, the pedicle valve. And so this is our brachial valve. And so what you see are some muscles. So there are what we call adductor muscles. So add means toward, and ADDUCTOR. That means the muscles that bring the two shells toward each other. So those are the closing muscles, adductor muscles. There are usually two sets of these, and they're going to connect from one valve to the other. So they'll hit the posterior part of both valves. And when they contract, they will snap the little brachiopod shut like a clam. Yeah. Now, the other muscle is the diductor muscle. And so this one's usually really wide. Sometimes it'll stretch all the way down to about here. And it reaches up and it just only makes contact, not in the middle of the valve, but right here on the cardinal process of the valve, of the brachial valve. So it's the very tip in the center. And it's just pulling on that thing. It's kind of like trying to open. You ever try to open a door by sort of grabbing onto the hinge and just like trying to pull the hinge to make it open? It's real hard, and it's a dumb way to open a door, and that's the way the brachiopod opens its shell. So from this, you can kind of get a sense that these things have two sets of really fast muscles to close, and so they want to be able to shut at first sign of danger. But they have one really weird, kind of badly organized muscle to open that's probably very inefficient. And so they're probably slow to warm up, slow to open, and real fast to close when they're feeling threatened. You can see these muscles on here. So they'll leave little scars on the shell. So this is a Sulcataria, actually, that I traced and got the insides. So there are our adductor muscles. So they're connected here. This is the brachial valve of the shell. And so it's got the prints of the lophophores. It's put on there the little maybe digestive area prints. And yeah, there's various other parts like the median septum. That's going to be our little wall to keep your water from touching my water between the two lophophore halves. And then up along the top, you can see a very thin thing called the outer socket ridge and a big, thick, wide thing called the inner socket ridge. And the hinge tooth will socket right down. It's actually more like that. It sockets right down into there. The space in between is the socket. This little cardinal process pit, which is sometimes called an antron, but we can call it cardinal process pit. That's fine. It is what became of that early fold in the shell during development that I'll show you. And presumably some organ or organs are back here. We don't know what the brachiopod's really doing with its cardinal process pit, but it doesn't go anywhere. It can't like excrete waste out of it because there's no hole. It doesn't open to the outside. So I don't know. All right. And if we go to the other valve, so this is the one that would be on the bottom, the ventral valve or pedicle valve. Here it's got, so this is the little notch where the pedicle would have come through if it's one of those that has a pedicle that comes through. And sometimes it'll have a little cover that's called a pseudodeltidium. So you see that on a lot of the shells that they have there in class. And at the back of that is like the, just sort of the bottom of this where all the other parts will attach. And so it's like a little platform, like a little diving board called an apical callosity. Yeah, and that's this blue thing. And you've got a little tiny medium septum, usually much smaller on the pedicle valve than on the brachial valve. And here, these giant brown tracks, those are the diductors. So they're starting out on the pedicle valve, reaching around, and they're going to be reaching up to the brachial valve, which, so here's our little cardinal process. There's a little groove on this side and on this side. That's where the tendons of those diductor opening muscles are going to lock. And so most of the muscles attach to this valve, but the little tendon will connect to the other valve and they'll pull it open just by reaching around the back. The adductor muscles will both line up right along the center here, some configuration like that. And they both come to roughly the same place, even though they started off kind of like that. All right. So now I would say pause the video, like they always say on YouTube, and have a look at these. See if you can figure out what's going on. Like take Echinaria here. Can you find the brachial ridges? I bet you can, right? See them? They start right here, curve down, and then disappear. Can you find the median septum that would be there to sort of deflect water? Oh yeah, look at that median septum. Can you find the adductor muscles, the adductor muscle scars? Like there's a set, like these little grapes hanging down, and there's the other set. It's kind of sort of finger-like up here. Yeah, and so those are the adductors. Can you spot where the diductor would connect? Well, the answer is no, because the diductor only connects, it reaches up through this little tiny groove here and this little tiny groove to make contact with just the end, which is just barely what we can't see. And it will mostly be attached to the other valve. These are all just brachial valves because they have more interesting features on them, I think. And so here are different kinds of brachiopods, and you can see some of them will have a long or a short median septum. Some of them will have different shapes of lophophores. Some of them will have all kinds of these little spines that stand up here that are after the lophophore. So it's pulling in water in the center, filtering it out over these things. And so I don't know if these spines are kind of pushing water back, redirecting the flow, or remember, there were probably tentacles on here that went out to pretty near the edge of the shell. So maybe that's what's going on there. You can see some of these have like little, it's like little diving boards where maybe the tentacles would have sat and come off. Yeah. So, yeah, now you can kind of read the inside of a brachiopod. I think the only other one I'd show you is this guy. So we've got nice median septum here. Here's our lophophore support. So this is the brachial ridge where the lophophore would sit. And then the adductors, there's going to be two right in here next to the center. There's a little ridge that divides them. And the other two are going to be back here. And they're all kind of somewhere between oval and triangular. You'll see they call that subtriangular in some of the papers, which I don't know. It's not any more of a triangle than an oval, but there you go. All right, so yeah, progress. And here, once again, this is like reimagining the shell, but with little fleshy parts on it. And so it should have some kind of lophophore, with the new growth being at this end and everything basically moving along as the shell grows. And there, you would expect the mouth to be right about in the center. Here are the adductor muscle scars. These are the anterior ones. These are the posterior ones. And this is roughly how we think they would have set in the sediment. A lot of brachiopods will have either spines coming out the back or spines coming out the hinge, and we think that's just like a comb to sort of plant them in the mud so that they can't get flipped over by the waves. Yeah. Here is a little thing showing development of the shell, because not all the parts develop at the same rate. So these are these are all the same species of shell. It's called Lysoconides primarius, and you find them at the finest shale in Jacksboro, among other places. You can see it starts out as just a little tiny shell with a great big groove here, and there's nothing in the groove. But if you look later, once it gets to sort of adulthood, there, it's developing its little median septum. So the median septum develops really late in these guys, like really, really late. All right, now let's look for these things. These little prongs here are called andoridia. Nobody knows what they're for. You find them in certain kinds of brachiopods, and it's like a marker that you're dealing with that one. It's some kind of little road that would have lifted up some sort of body part higher than the rest of the body. Did it go to the gonads? Were they upstairs for some reason in this version? Are the kidneys sitting on top of these? Are they, I don't know, separating the lophophore from the digestive area? No idea. No idea whatsoever. But you can see they're here in the adult, and here's one there, and here they are here. And you can see just a little bit of them here. They're just barely starting. But in the slightly earlier version, those don't exist. So that's a feature that develops, it's kind of in the middle of development. And you see it develops more of these little rows of little spikes on the inside. You can see it has tons and tons of rows, maybe 20 or 30 by the time it gets to full size. Whereas a baby, it starts out with one row on each side, and then it just keeps adding them as it goes on. It starts in the center, adds to the outside. And you can see the groove in the center. This whole area of the groove is going to form the cardinal process pit, which there's our pit, there's our pit. And then this other part of the groove down here is what will eventually widen out in this big, look at that, big triangular sort of groove. And that's where the median septum sits. And so this particular kind of brachiopod was really into having a very wide triangular groove. We don't know why. I don't get what that gains the brachiopod, but darn it, it's doing it, and there it is. Yeah. Okay, I think the last topic for this section is the shell. So how do they make the shell? Well, this is from a paper that's the genome of Lingula, which is this brachiopod up here. That's the baby of it. And it's the one that has like the massive long pedicle, and it's sort of a long oval shape like that. Yeah, that I've shown lots of pictures of. And so without going into all the details, because they're in the paper, but yeah, I don't know that you need those right now, the way they build shells is that they are going to lay down a layer of chitin, just like an insect armor, and just like the first layer that's laid down for mollusks. So you make a little chitin floor mat, basically. And then on top of the chitin, they're going to have lots of different proteins, and these proteins are going to bind things like metal ions or calcium. And so if you have a calcium phosphate shell, they'll bind calciums or calcium phosphates. And these proteins are going to start out the process of shell building. They'll sort of lay down a couple very carefully orchestrated metal ions. And then from there, yeah, the crystal will just grow as long as there is calcium carbonate or calcium phosphate in the area, and the brachiopod will keep pumping it into the area. Then it'll just crystallize on the outside of these. So the proteins form like a nucleation point. It's kind of how the crystal starts growing. If you look at the color coding, I think that's kind of neat. So there are in Lingula, the brachiopod, there are some things that are specific to Lingula, which good for it. But you find a lot of proteins. All these things in green are conserved, and you would find them in mollusks, and they're used to make the mollusk shell. And you would also find them in bone, like our bones. We have the same proteins, and they're part of the same process that lays down bone. So this is a very similar sort of process using conserved machinery. Over here, these orange guys are things that you would find in both mollusks and brachiopods, some brachiopods. And the blue things are things that you would find only in bones and this Lingula brachiopod, some brachiopods, but would not find in mollusks. So it's not like mollusks give rise to brachiopods, give rise to people. It's just that all three of these things are sharing from the same gene pool and stealing the same sort of genes from a common ancestor or from each other and using them to build protection. Yeah, that's what bones do. They're sort of protection on the inside that lets us move around. And they can build protection on the outside, like a clam shell or like a brachiopod shell. Now, last slide in this section. If you look at the really, really early shells, so bottom of the Cambrian, these are things that are probably brachiopods, but there's little differences that make you say, I don't even know if these are brachiopods. They all have the same shell construction, and this is something you see carried through all the way to modern brachiopods, except the modern brachiopods kind of fill in the spaces between these. So the way it works is you put down a primary layer, which is this little PL right here. The primary layer will have little holes in it for those sort of sensory setae to stick out. So it can tell if something is coming up and going to eat it, and then it can sort of retract lophophore, close shell, go into a full little brachiopod panic. And these little tubes are there only on the kind of growing edge, and they quickly get basically paved over from the inside, because remember, our shell is going to grow from the inside and just toward the front, toward the anterior. Then in the growth regions, you're going to have these columns. And so there are probably proteins that are directing the formation of the column and building a shell very carefully. And then you get to the lamina, and the lamina are probably the layer where you have all the chitin. So we'll probably put chitin down on the bottom and then grow our little shell with some of those proteins that are nucleating calcite. And brachiopods got much better at this. That's why this area tends to be totally filled in with calcium carbonate in most brachiopod shells. Then you got your lamina, and then you got another layer of columns. So it's, column and lamellar architecture, if you like. It's what you find in brachiopods, then and now. And if you were to slice brachiopods open, you can see these little columns, basically. And you can see the little holes where the setae used to be poking out, but where they were sort of closed off from the inside so that they, wouldn't be poking out anymore. All right. Hope that makes sense. Yeah, you got a real good picture now of like how brachiopods work, I think. And so the next part, which I think is the last part, let's just look at some of these and get a picture of their overall evolution, because I think that's kind of fun. All right, see you then.